Good afternoon, everyone. Thank you for coming. I'm not sure how many, we have another 15 people online. This is actually quite a good turnout for seminars that we have here, so thank you for coming. So this speaker, Giovanni Parmigiani, is a very special person to me. Just to state my, what's that word, my conflicts of interest. When I've got into bioinformatics at a later stage as a postdoc in Toronto, and then my second postdoc, I was fortunate enough to get in at Dana-Farber Cancer Institute and Harvard School of Public Health, where Giovanni was one of my supervisors. So at the time, he was a professor of biostatistics in the Department of Biostatistics and Computational Biology, and doing work in various areas, including Bayesian statistics, including gene microarray analysis, various areas of genomics. He has always had a fairly broad range of research areas, but always with interesting statistics and a very practical approach to them. So now I believe his title is slightly different, and director of some institute, but doing, it looks to me, fairly similar things. What I would encourage everyone to look at here, and whenever I have had the chance to read Giovanni's papers or see one of his talks, I would see it not just as this is about risk prediction in cancer, but this is also about how do you take an important problem that you're interested in, take the data that you have available to you, and find a way to address that problem in whatever way works. And I have always appreciated the very practical approach that Giovanni takes to problems, whether it be borrowing things from the Bayesian world, the frequentist world, from the computational world, from clinical trials, anywhere where there are data that can help you to understand something about your problem. So, and I will also say that, to the extent that all of you know me, I would say that Giovanni has had an enormous scientific imprint on me, and I have tried to, I learned a lot during that 3 years, and it has continued to influence the way that I do science and talk about work with other people and advise and all of these things. So it's really a special moment for me to have Giovanni come back here to where I'm working now and to come and be our speaker. So I don't know, it makes me emotional, and I'm very happy to have you here today.
Thank you, Levi. It really means a lot to me. Levi is one of my favorite people in the world, and he knows it. He was also one of the most influential trainees to ever participate in the research in my group, and I have a very long list of trainees and very distinguished one, I'm proud to say. But the work that he did with us created data infrastructures that we still use to this day, both in research and in teaching, and many Harvard students are growing through the pains of looking at the curated ovarian data. But also, more importantly, conceptually, some of the way he stirred us towards thinking about multi-study learning and cross-study validation of prediction models was truly unique, and it really has influenced the way we've done things for many years after he left the lab, and to this day, including several grants that are ongoing. So that's great. And Levi has also taken an interest in my old digging places and in our country and the beautiful mountains thereof, and so that we have even more in common now than we had back when he was at Dana-Farber. But we haven't been seeing each other for way too long, so today is truly special. It's great to be back. Great to see him doing so well. Great to see him at the helm of a group that does wonderful research. Truly special. Pleasure to be here, and thank you very much for the invitation. I want to make sure I don't go too far over time. I've prepared enough material for 6 hours, so I'll have to. I was kidding. I definitely hope not to speak until 5:30. Let's see. Very much. Yeah, here we go. I think maybe this. The other way. Not this.
I just minimize this one. Can I get this? Okay. Okay, there we go. And this one, move that out the way.
Okay. Old-fashioned way. Drag it out of the way.
Okay. Yeah. There we go.
Okay.
All right.
I'd like to open with this with a slide of disclosure information, but I think the only one that is going to be potentially somewhat overlapping with today's discussion is an ongoing research collaboration with Ambry Genetics. I don't derive any personal income from it, but there's a lot of data and computational stuff that is part of this. And also I will describe some risk assessment tools that we do license to both healthcare providers as well as software companies that do sell to healthcare providers. But all of the licensing comes to the institute and goes back to the lab for software maintenance and upgrades, and there's no personal income associated with that either. So I have two parts of the talk. The first is on high-risk families and cancer and risk assessment in the context of that. And it's a topic that's been very important to me for personal and academic reasons for about 30 or so years. And we've been working in this space since BRCA1 and BRCA2 were found in the mid-90s, and I was working at Dunbury and then onwards. It really means a lot. And I'll show you the general framework of how we have been thinking about risk prediction in that space, and then a couple of the latest developments from both a methodological and an applied standpoint. But then I wanted to squeeze in a part two where we zoom way out from the context of cancer and high-risk families and inherited susceptibility to looking at risk prediction more broadly, in the context of everybody, and looking at multiple diseases at the same time, not just cancer, but looking at the chronic disease evolution across a variety of diseases using electronic medical record. That is much more recent work that has emerged out of my interest in latent variables and matrix decompositions, which actually is also another thing that I owe you all, Levi, a big debt of gratitude. He's the one who stirred me to try to think about whether different latent variables that we learn from things like factor analysis of single divisor decompositions would be replicable across study, a stream that I took very seriously, and we've done some really fun stuff as a result of his initial idea. I imagine most of you will be familiar with the context of genetic testing for inherited susceptibility to cancer, but just as a way of a quick review, cancer clusters in families. It clusters in families as a result of the fact that relatively high proportion of people inherit defective copies of important genes from their parents in a heterozygous type of way. And the effects of that are only seen later on in life, past reproductive age. So these genes are still very much part of the human population. Famous ones are BRCA1 and BRCA2, PALB2, et cetera, for breast and ovarian cancer, as well as the genes that are associated with, say, the Lynch syndrome. Those are genes associated with defects in mismatch repair. And they tend to cause colorectal, endometrial, and other types of. In addition, there's now hundreds of such genes that have been identified through various types of genetic epidemiology and molecular biology analysis. What happens to someone who carries a defect in one of these genes is that when they're born, every cell has only one functioning copy of the gene. Say, let's say it's MSH1, a gene that is part of the machinery that does mismatch repair. Then when that cell loses if and when that cell, one of the cells loses the good copy, then the cell acquires the ability to mutate a lot more easily, and that becomes one of the driver mutations for the development of cancer. So in the sort of two-hit model of cancer development, the folks who carry genetic susceptibility are one step ahead of everybody else in that all of their cells have an increased vulnerability to the double hit. So as a result, you see very clear Mendelian patterns of inheritance within families and clustering of cancer. The strength of this effect varies depending on the type of mutation that is inactivating the gene, as well as the specific gene, and it's not a definitive sentence, in that many people who have these mutations live perfectly normal lives. So there's incomplete penetrance, to use a genetic epijargon. And there's quite a bit of variation in risk. There is also quite a bit of cancer that clusters in family as a result of shared environment, shared behavioral aspects of people's exposure and so on. And so disentangling the genetic susceptibility from other sources of susceptibility is not entirely straightforward, but it is important because to some extent in the US, it is still required to have a sufficiently high probability of carrying one of these genes for the genetic testing to be reimbursable. And also prospectively, the consequences of having a genetic susceptibility are very different in terms of the implication for what one might do in terms of screening and the implications for one's relative. So in this context, there's quite a bit of interest in assessing both the pre-testing risk of carrying one of these genetic mutations and the post-testing risk of developing cancer, given that one of these mutations is found, or sometimes even given that a mutation is not found, or there's a third and even more complicated path that happens when a mutation is found, but the relevance of that mutation is of unclear clinical significance. So this is represented a little bit in a cartoon way in this general picture. Typically, genetic testing is the data it generates tends to focus on families at high risk and collections that are at increased where cancer clusters in the first place, which is an additional challenge to many of these studies. So I told you about a few examples of the genes that are important. These days, it's very common to test for very large panels of genes. And the point of this figure is not to focus on any of the individual specific genes, but it's just to show you how rich the landscape has evolved to be. Each row here corresponds to a cancer susceptibility gene. Each column corresponds to one of the major cancers that happens to be listed in this table. And a dot is presence when there is a known susceptibility to the cancer in the column associated with the gene in the row. And so it's quite a complicated landscape. Just keep that in mind as we proceed to the discussion. I'm going to now take a step back and focus just on a single hypothetical point in that matrix, a point that connects the row corresponding to a gene called PALB2, which is a partner of BRCA2 in forming a protein that is part of double-stranded DNA repair, and ovarian cancer. for a very long time, it was unclear. It has been unclear whether there was an association here. In the table that I showed you earlier, the association is now marked as present. It's still very difficult to estimate the risk. We are right in the middle of doing a meta-analysis of the studies that investigated this risk. There are hundreds of papers published on this. Most of them are on relatively small populations. The statistical significance of the result is inconsistent across these various studies. The design is often fraught with substantial ascertainment challenges because of the way the families become available for testing, and that depends on whether or not someone has cancer. So you're essentially stacking the deck in favor of seeing an effect in some circumstance. And there's also quite a bit of heterogeneity in the way the risk itself is reported. Ultimately, if you are counseling someone about their risk of ovarian cancer as a result of a PALB2 mutation, and that person is 40, it's really important to have a risk that depends on age, where you can have you have the ability to give them a sense for what is the risk of developing ovarian cancer in the next 5 or 10 years compared to just a lifetime or a relative risk compared to the baseline. There's an age of onset shift that is often associated with carrying this susceptibility because of the 2-hit model that I was describing. It anticipates the whole timing of cancer evolution, and there's also a fundamental importance of knowing about the age because of the type of decision making that is involved. In this case, often it's the oophorectomy, and so there may be decisions about family life that are involved in here. For which it is important to have a curve. This curve is not always available from this study. Sometimes it's only a relative risk or an odds ratio that is available. So these meta-analyses are quite challenging from a statistical standpoint. And if you go back to the previous picture, you have to multiply this by each row and column. So it's quite a complicated task. It is, however, a very important bottleneck in the translation of everything that we are learning about these genes and these populations into what the genetic counselors can offer in terms of decision support to the patient. So our mission has been to essentially use statistics to facilitate this translation of this whole knowledge into something practical for genetic counselors. Yeah, this is more of a cartoon of pretty much what I've told you up until now. It's very difficult to have comprehensive studies where you can estimate carrier probabilities for genetic susceptibility and subsequent risk based on single data sets, although we are beginning to get there, and maybe that's the future. But for the past 20 or 30 years, we've mostly relied on a patchwork of contributions to the literature. So that's where it starts. And one of the challenges from a statistical machine learning perspective has been to understand this literature, both from the standpoint of identifying relevant studies, classifying them by quality of the design and relevance, and then curating the content in a way that it becomes computable on, and then doing meta-analysis. So these 3 major steps: the classification of relevant paper, the curation and extraction of the relevant information, and the meta-analysis of the final results have been sort of places where progress in machine learning has been lagging, and we've been trying to develop development methodologies and apply them. The final result, ideally, is a risk estimate that is associated with carrying a specific gene that is absolute, that is, it's a probability of developing a specific cancer within a certain interval of time. It's not a relative risk, it's not an odds ratio, it's just something that you can hang your hat on in terms of medical decision making. It's personalized in terms of the fact that it depends on the specific gene, as well as potentially other risk factors or even aspect of the family history can be relevant. And it's personalized with respect to the age of the person. So you would give a different prediction to someone with PALB2 mutation and no ovarian cancer who is 55 compared to someone. So that's some of the that's where we want to arrive at. I'm going to show you one example of one of these meta-analyses that is one that is kind of a middle of the road in terms of challenge and high throughput one in terms of how many studies there are and the quality thereof. It's a meta-analysis that looks at MLH1, MSH2, and MSH6. These are 3 genes associated with Lynch syndrome, and they all have to do with mismatch repair deficiencies. And we gather all the studies available. We have a model that surmounted the difficulty of studies reporting different measures of risk. Also, different studies aggregate the 3 genes, or sometimes they provide separate estimates. They aggregate gender, and they handle age differently, sometimes in an aggregate, sometimes in a disaggregated way. Each study has different ascertainment mechanism. The people are included in the study based on different criteria, and the quality, the resulting quality of the study varies as a result of that. So we have concocted a statistical approach that can account for all of these various methods, and we have applied it to a paper that appears a couple of years ago now in what's this paper? In JNCI on what I forgot the name of the online version of JNCI. So in this figure, what we represent is for each decade of age, each of the estimates from the various studies is aggregated by males and females, coded in as blue or orange. So you look at this figure in blocks of 10. The meta-analytics results are in bold for each of the 2 sexes, and then they're smoothed through using a parametric fitting, which is ultimately what then gets fed to the decision support system. So here it's a little bit messy and crowded, but you see every bit of information that goes into that decision support approach. Each ingredient to the meta-analysis, the difference by gender, the meta-analytic estimates, and the smoothed version of the meta-analytic estimate, which is what allows us to personalize the absolute risk to the exact or the age in years of the person that is being counseled at any given. These estimates are used as you described, as you've seen, straight up to give decision support to folks whose mutation is found to be one of these. But there's also an aspect that I alluded to earlier of identifying people who are likely to carry these mutations prior to the genetic testing. And these type of studies have a role in that as well, because they serve as the input training parameter to a family of prediction models that we call Mendelian model. I'm going to explain to you very briefly in the next couple of slides, that are designed to take this type of risk by age, as well as information about the population level allele frequency of these mutations, and produce a personalized estimate of the carrier probability for an individual whose pedigree you know. This pedigree is actually from an old article that appeared in the New York Times where they interviewed this person, Deborah Lindner, whose family has a long history of breast and ovarian cancer and who carries a BRCA1 mutation herself. And obviously, as a person who had agreed to be in the New York Times, she agrees to this information being shared publicly. But it's a really interesting article to get a sense for all the various implications of being a carrier of this type of mutations for herself, her family, her sisters, and so forth. Anyway, here is to serve as a reminder to recommend you to read that article, which has been around a while, but it's quite interesting, and also as a placeholder for the individualized part of the input to this model. So at the top is the training part, the part that is bringing in the population level information, and here is what a person would bring into the genetic counselor's office. It's not used to retrain the model, it's just used as the vector of covariates, if you'd like, that is used to calculate the prediction at that. And then this gives you a probability of inherited susceptibility given family history, and I'll make that a little bit more clear in terms of exactly what type of genes are considered. So a couple of slides on how these Mendelian models functioned. It's a long history. It goes back to some folks who were at Johns Hopkins in the 60s, Murphy and Mutalik, a statistician and a geneticist, who laid out a very simple approach for using Bayes rule to calculate the probability that an individual carries a particular allele given their history of phenotype in the pedigree. It's the logic is relatively simple, and it is essentially looking at the pedigree as though it from a statistical standpoint, as though it was a Bayesian network, and it applies Bayes rule in a chain type of way. I have a little bit of a cartoon for that, which I drew on the board one day and then took a picture, and just always. I think it's the first thing I ever drew on my office at Dana-Farber when I got my new board. So I remain fond of it, and I still show it in this kind of very primitive format. But this is the network representation of a pedigree where you have two individual grandma, mom, and her, which is the person currently sitting in the genetic counselor's office. In the squares, you have the quantities that are known as typical in this type of Bayesian representations, and H represents the health history, whatever it may be. It's very often consists of maybe the age of whether or not the person has cancer, if they had cancer, the diagnosis. In the case of breast cancer, often the subtype. In the models where we look at for example, a pancreas cancer, we're also interested in whether the person was a smoker, that sort of information. It's all condensed into the age. Before genetic testing, the genotypes of all these individuals are typically unknown. And so the circles represent the genotypes. G3 is for the counselee, G2 for the mom, and G1 for the grandmother. And the arrow represents the relationship of conditional independence between these nodes. So the typical Mendelian models that are used for Mendelian diseases with strong effects assume this type of conditional independence, where for example, if you look at mom, the mom's gene depend on grandma's gene, as is obvious, but mom's family history, mom's history of disease does not depend directly on her own mother's genes. It depends on those only through the fact that some susceptibilities may have been passed on to her. So while in contrast, H1, which is the health history for grandma, depends directly on those, and so forth. This obviously does not account for the possibility that both mom and grandma may have been exposed to the same carcinogen because they were both living in an environment that was rich with that. It only is useful when those effects are relatively minor compared to the effects of the gene. But as far as major susceptibility genes for cancer, this way of modeling has gotten us a very long way because it is so modular. It allows us to break down the problem into the point where we can do the entire modeling of all the unknowns in this graph just by looking at the allele frequencies of the founders and a likelihood model for phenotype given genotype at the single individual level. And you can extend this logic to pedigrees of any size as we have, to genotype of any length and we have, and to family histories that are quite complex and we have. So the simplification of not looking at non-genetic aggregation here has enabled us to break down the problem into smaller pieces and populate each of those links with information that is available in the literature. Sometimes we do a little bit of a Frankenstein job of putting together things that are coming from very different sources. But our approach has been going throughout the years is that we try to bring in the best information as we can, cook up our model, and then do an external validation to see whether it works well enough to be better than what the clinicians have in hand at this point in time. So there's a lot of technical stuff about this that I would enjoy getting into more detail, but I'm going to move forward and I'm going to tell you what is our latest incarnation of Mendelian model. We have originally developed a model called BRCA Pro that was published in the late 90s and has supported genetic counseling in the breast ovarian space for quite a long time. That model looked at breast and ovarian. You see breast here and ovarian here, and look at two genes, BRCA1 and BRCA2, and that's why those are in the orange here. Then years later, in my Hopkins days, we developed a model called MMR Pro that looked at the three main genes from Lynch syndrome and colorectal and endometrial, and that's the orange here. And then we also developed a model for skin melanoma, and the gene that we looked at for that is CDKN2A. Over the years, as a result of our interest in remaining relevant in a world that is the world that I showed you earlier, where we have panel testing with as many genes and cancers as I've shown you, we've been trying to use the approaches that I've talked about up until now to scale this up one bit at a time. And we're now at the point where we have the cancer and gene combination that you see here. It's a relatively small number compared to where we need to be, but it is at least covering the panel testing for the smallest panels that are commercially available from the main genetic testing companies, and it's covering a relatively wider array of cancer type. Each of these squares is a meta-analysis like the one that I showed you for the colorectal cancer, unless there are studies out there that we can directly lift the penetrance estimates for with trust. So that's where we are now. This requires that the gene variable in that my scribble on the board is a vector of this dimensionality, and each individual can be heterozygote or homozygote potentially in two ways, because in some of these genes, heterozygote carriers are also viable. So it's 3 to the power of that number, a number of possible genotypes that we need to consider when we integrate it out. And for those of you who have ever used peelings or something like that, you know what that entails, but we have surmounted some of those computational challenges, and now we can give this prediction in real time in a way that is relevant for genetic counseling. So yeah, I want to show you this slide, which I think it's from a paper that is currently being under review about the external validation of this larger model. And it compares it to the BRCA Pro and MMR Pro models that I just told you about. On the X scale, you have the probability of carrying an inherited susceptibility mutation in any of the gene considered by the corresponding model. So here would be the probability of carrying a mutation in BRCA1 or BRCA2. And on this one, it would be the 3 MMR mutation genes that I told you about. On the Y scale, you have the corresponding probability for the bigger model that looks at the ensemble of genes. And you can you can see a diagonal here for folks who have a strong family history, in this case, of breast and ovarian cancer, but also something that shoots straight up, meaning that you have a very small probability using BRCA Pro and a high probability using this more comprehensive model, which we now call FAM3 Pro, including some that are pretty high. Now, some of these are individuals who have a high probability for the mismatch repair genes, but many others, the ones in black, would have fallen through the cracks because they would not have been detected by either one of the two models. And so the black points that are off the diagonal and have a higher probability than the diagonal in both of these figures are the value added of looking at a more comprehensive way. Clearly, the earlier syndromes were identified with the genes that are more common. And so in terms of overall frequency, the new genes that are added are more rare, and the overall public health impact of the individual genes is small. But overall, they can still contribute quite a critical mass. And very often we find that we face situations where a family has a very clear inherited susceptibility, and we don't know where the hell that is coming from. And I really feel strongly about being able to provide some guidance in terms of explanation and not just screening recommendation to those families. I have one more validation slide. Yes, I don't want to dwell too much on this, but despite the fact that we've added so many genes and so many cancers, we remain overall fairly well calibrated in terms of the proportions of carriers that we predict. These here are calibration intervals by gene for 3 different cohorts where we applied this model. The 3 cohorts are labeled by both color and the different symbol. So there are some genes for which we are observing more than we are predicting. For example, for CHEK2 here, we observe about twice as many of the carriers as we predicted, but by and large, we seem to be reasonably well calibrated. By looking at so many genes in cancer, we lose a little bit of resolution on the more rare genes, but compared to our earlier models, we're still pretty much on the same ballpark in terms of the genes that are associated with the major syndromes. So I think that this is ready for clinical deployment without any loss of accuracy compared to the older models that have a smaller scope, and with a reasonable ability to capture new cases for the genes that are a little bit more rare. Now, I'm hoping also that this problem will go away, that genetic testing will become so much cheaper that there will be no need for being so sort of machine learning about giving people carrier probability before they can be tested. But I think that this model will retain an important role, particularly for the forward risk prediction. I want to wrap up this part of the discussion by showing you a patient-facing tool that we've developed using the Lynch syndrome meta-analysis that I showed you a little bit earlier. This is something that is live on the Dana-Farber website, and the risk communication that we do is to some extent adaptable to a patient's own preferences. There are 3 different ways for patients to get the risk. Some are more qualitative, some are more quantitative, some are age dependent, some are lifetime. You can choose your interval and get we can give slightly different presentations of the results to the patients and the genetic counselor who may be more quantitative or may want to do some reprocessing. All of this is the result of some focus group that my lab co-leader Danielle Brown has done with both the genetic counselors and some high-risk families to try to figure out what is the best way to present the information. We also present information about potential screening strategies and intervention, diet, aspirin use, colonoscopy use, risk reduction associated with colonoscopy. The risk reduction associated with colonoscopy is coming from a completely external set of information that is not part of the Mendelian model itself. And I was having an interesting conversation earlier about this. Maybe in the future we'll get smarter and be able to handle this as part of a single modeling effort. But that's the way things are represented so far, so that patients have counterfactual scenarios. This is what your risk of colorectal cancer will be in the next 10 years. Just if you don't do anything, this is what happens if you do screening annually. There remains the challenge of the additional possible cancers that are associated with this syndrome. And I see all of these tools as good ways of concentrating a lot of information that is used for decision support, but none of them can be considered as a definitive in any sort of way, because the knowledge is accumulating and we're getting more information about these populations and about the effects of these genes. I mean, if you think about it, the expressivity of this deficiency in colon and endometrium is strong, but there's also a long tail of consequences for a number of other disease sites. The entire GI tract is involved. There's some evidence of increased risk of brain cancer. And in principle, the deficiency itself is a mismatch repair deficiency. It means that when your cells get hit by a carcinogen and there's a single nucleotide substitution, the mechanism that normally repairs that substitution is going around without a spare wheel. And so for the cells that then lose the heterozygosity at that particular gene, those mutations will not be repaired. And that can happen in any tissue. Now, obviously, a tissue like the colon is right in the line of fire of a lot of exposure to different carcinogen is reasonably more likely to develop cancer as a result of this. But there's really no reason in principle why any tissue should be at baseline risk once a mutation like this is. So we want to continue enriching this with additional information, and it's a fine line both in terms of how reliable the information is and how big the risk is, because it's quite complex to fathom the fact that you're now at risk of 10 different cancers with such a different spectrum of possible things that you could do to prevent them or to catch them early. So it's a really complicated space to navigate. And the science and the decision support need to go hand in hand, but there also needs to be sort of a reasoned approach at providing information that could be overwhelming and maybe counterproductive. Quite complex. All right, credits for this. Most of my credits go to Danielle Brown, who leads the lab with me. These are some of the papers I showed you. Also, the first authors. Kathy did the meta-analysis of the mismatch repair gene. Jane Liang is really the mind behind the FEM3Pro paper here and here with the genetic, with the statistical methods, and Gavin wrote up together with Jane. an earlier version of this that used to be called Panel Pro. Then our technology transfer office told us that Panel Pro is some software that they use to control your irrigation panel from, so we could not use the name for some reason. We had to change it, and now the new name is anyway. Lots of people to thank for. A lot of the validation were done in collaboration with Steve Gruber and Greg Ides at City of Hope over in California, and they're great to collaborate with, and they have really good populations. Okay, so how am I doing with time? Even my little mini number over there has disappeared. So 4:50. Okay, so I'm going to give you a 10-minute overview of this completely different project with a very different flavor. The data is going to be electronic medical record data. The goal is going to be to capture many diseases together, and so those are two sort of major changes in perspective. Motivation, develop multiple diseases. Everything you've seen so far is very focused on specific condition, and it works in the case of strongly Mendelian genes, because that the consequences of that should be on top of a person's list. But there are many other things if the risk is lower, they should be considered. The other important thing is that disease comorbidities, disease co-occurrences follow patterns, but these patterns vary by individual, and they vary within each individual. They vary over time, so they tend to be a bit hard to capture. And genetic influence is important, and we need to understand it, but we need to understand it in a probabilistic way, just as in the example that I've shown. So the approach that I will show you takes the perspective of looking at signatures as a way of capturing the underlying biology of diseases that occur together. So a signature would be some underlying propensity of developing diseases of a certain type, for example, cardiovascular disease. And it might be associated with different diagnoses that typically come up with cardiovascular illnesses. So if we are going to train this model as we are on data based on electronic medical records, we may have multiple diagnoses that are associated with cardiovascular conditions, hypertension first, and maybe angina, and then later on congestive heart failure or atrial fibrillation. And these may be associated with the same underlying process of aging or cardiovascular illness, but they may manifest themselves at different times. So what we wanted to have is a description that is time dependent, that is age dependent, of how the different diagnoses contribute to creating these underlying traits. We also want to be able to understand the genetics directly in terms of the effect that having, say, a certain genetic makeup, or let's say it's captured by a polygenic risk score of some kind, how does this affect not the individual diagnosis, which is a very noisy problem, but the latent variables that are formed out of this individual diagnosis that describe, hopefully in a more substantial way, the underlying process, with the idea that that relationship is more likely to be more robust, easy to estimate, easy to identify, and more productive in terms of the ultimate prediction. So to arrive at that point, we cooked up this really complicated model that is essentially a matrix factorization, where the factors or signatures are allowed to vary over time for the reason that I described. But also, each individual has his history is determined by which signature they are quote unquote part of, which will be defined probabilistically with weights of some kind. But these weights themselves will change over time. So that if you have an early onset of, say, a cardiovascular condition, but then you start taking statins and that becomes less relevant, that is allowed to kind of wane, and then other things may take over. So we allow both the signatures to be age dependent and the importance of those signatures for the individual time history to be age dependent. It's quite a complicated model, and it's semi-miraculous that we were able to fit it, to be honest. And the credit goes to mostly to Sarah Urbert, who is a cardiologist, but who has an MD PhD in genetics and an undergraduate degree in statistics, and she can do everything, absolutely everything, and was able to integrate. Because to fit a model like this, you obviously need a statistician, you obviously need a geneticist, and you obviously need someone who understands these codes at a much deeper level and sees the patients and sees whether the results make sense from that perspective. But even an interdisciplinary team of that sort would have a very hard time keeping all the communication needed to pull a project like this. But she's the one person who has all that expertise, and so it's been miraculous working with her, and she's constructed something that's a thing of beauty. And yeah, so the training goes that we have N individuals, for example, the whole of UK Biobank, D diseases, for example, every diagnosis in the fee codes that you can derive from the UK Biobank. That's thousands. T time points we are doing, I think, yearly or monthly. Then case signatures, we have 21 of these disease classes that we worry about, and then we have polygenic risk scores that we put in as covariates at birth, along with sex and a few others. So the data is this three-way array, or tensor, if you want to be fancy, that is by individual disease and time. And for each, we know whether the person was diagnosed with that disease at that time. So this giant Bernoulli tensor that we try to decompose, and we decompose it in signatures that are time dependent. Those are called lambdas, and in the importance of these signatures for each individual, those are called theta. I mean, maybe a cartoon will be a little bit more effective. So a signature at the population level, for example, for the cardiovascular, in this cartoon, it has 4 different diagnoses associated with it, and each diagnosis has a pattern over time. And then you have maybe a gastrointestinal pulmonary and something. Then for each individual, you see what the various diagnoses have been, and you learn the various weight for the signatures at various given time. If you truncate the history of an individual at some age, for example, 50, no, in this case, 30, you can also use the same model to project forward the importance of the signature for this person going ahead in age and do prospective risk prediction. So we've published so far a model called MS-Gene, which looks at everything up until the prospective risk prediction. It's in Nature Communications. It's essentially a topic model. It looks at the diagnosis and it tries to interpret it in the context of these quote-unquote topics or signatures to see how they cluster together. And then it associates the polygenic risk scores to these topics that are kind of more meaningful than the individual diagnosis, and certainly easier to analyze in terms of the genetics. And then the work that we're doing right now is to transform this into a prospective model for future risk prediction. I'll show you some of the ROC curves, and they look very promising, but it's very encouraging to me that even in a space like oncology, where there are very well-established models, we are able to do, say, better than the Gale model or better than models that predict pancreas risk in the specifics of that, because we can leverage comorbidities across the spectrum in a way that other models cannot do. Also, very important to me, we can give counseling that is different to someone who has a predisposition to cancer and a serious cardiovascular or pulmonary condition compared to someone who doesn't. Right now, what do? We do essentially what's called the net penetrance. We give a prediction that is assuming that we could remove all other causes of disease from someone's history, right? And then the doctor or the genetic counselor in the back of their heads does a calculation or a discussion with a patient that is different depending on whether that patient has diabetes or not. This goes beyond that and tries to incorporate that as part of the model. Okay. Yes. 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 That's ultimately the goal. I mean, I've showed you that we've done it in that much smaller world, and it took us 30 years, but we have the right models. We know how to populate them to some extent, and we have the patient-facing tools. So now we're jumping to a much bigger task, obviously, and I cannot tell you when we'll be in the clinic with this, but Sarah is unstoppable. So it certainly is going to take her a lot less time than it took me to to be successful with this type of work. Right. So these are four signatures. The one on the top is cardiovascular, and the value that I'm showing you are the time varying weight that each diagnosis has in describing the signature. So the gray ones are the ones that are not so important, and the colored ones are the ones that are sort of defining the signature and associated more specifically with that. In the cardiovascular signature, the ones that are important to it have pretty much a very similar time course. In other signatures, that's not the case. For example, here, the blue diagnosis happens to be much earlier than the rest of the diagnosis for the same signature. And it varies. The model learns from the data, and it's trained one way or another. These are in the log scale, so differences here are super important in terms of probability. Differences here are much less. Couple more things. Yes, this is a real version as opposed to a cartoon version of the retrospective analysis of a patient's history in terms of these signatures. And you can see here a cluster of diagnoses are super hard to read here, but they're mostly of cardiovascular disease. That pushes up the cardiovascular disease importance of that signature for that patient specifically. So before you were looking at the recipe for making a signature, now you're looking at the importance for this individual of each of the signature, given that the recipes are written up somewhere else. So for this person, the cardiovascular signature became very important around this point, retrospectively in hindsight of their entire history. And then later on, I think this one was Budenitis, I think, and someone else related to GI trouble of some sort, and that signature went up later on in life. Presumably, after this cluster of diagnosis, there has been some treatment of cardiovascular conditions for this person. This is not quite age, because I think we are enrolling at 30, so this is age plus 30, so this age is probably 56. But that's just an example, and if you look at this prospectively, so if you look at this person here, you wouldn't have this jump up because you wouldn't in hindsight know that these diagnoses were coming. But you can still calculate prospectively what the various risks are. And in this case, at this point, would be pretty useless one. It will only depend on the PRSs at birth because there have been no diagnoses up until that point. But in other scenarios where some of the diagnoses are accumulating a little bit more progressively and you start accumulating maybe diabetes or maybe other conditions that are more revealing of potential future participation to other signatures, the dynamic prediction takes a more sort of nuanced flavor. These are the ROC curves. There is no, we don't know of any model that does this out there in a prospective way. So we build a straw man using a Cox proportional hazard model to try to see whether at least we would beat sort of a reasonable benchmark, and we beat it in pretty much most of the diagnoses that we have tried. I'm super interested as the next step in having sort of a more specialized version of this for oncology, because UK Biobank is not super useful on this front. But I think we have promising preliminary result in discrimination in terms of the oncology space, as well. Last one is calibration. This is external calibration on set-aside data. And I think we do a pretty good job of that. I think calibration is something that, because we are so into absolute risk and telling people you have a 7% risk in the next 20 years of developing X, I think we really owe them some calibration through an ethical demand of these models. They may be low resolution, but if I have a collection of people and I tell all of them 7% of you will get disease X, it's important that 7% of them will get it. So we care about calibration, and we try to tune the model so that it is, and this is calibrated in set-aside sample in rotation. We'll following Levi's teaching, we'll also look at cross-study ability to keep the model calibrated as soon as we have the opportunity. So yeah, so Sarah is really the driver behind this, and she's a cardiologist and starting her own career, but she's collaborating with myself and the lab of Pradeep Natarajan, who's a geneticist mostly interested in cardiovascular disease, and the lab of Sasha Gusev, who's also the Dana-Farber. He's a genetic epidemiologist who is just another wonderful guy to work with. So yeah, so hopefully this will keep us busy for a while. I'm super excited about it. Hope we can push through a path to clinical application for this in much the same way as we've done for the cancer susceptibility. So thank you very much for listening, and feel free to ask any question you'd like.
Questions here, and I will turn the screen on here so that we should be able to see questions coming from online as well. If you're online, you can either put it in the chat or take your mic off when I call you, and I don't know how to do this. Somehow we'll there we go. Okay, we can see the chat if you put something here. Okay, yes, Rebecca.
A lot of personal reasons as well as just broad applications. EHR data depends on people going to interact with the healthcare system. How does this maybe suffer from that kind of selection, where people maybe don't seek that?
It suffers hugely from this type of selection. I mean, the model is trained initially on UKBB, and they have a sort of a centralized system. I don't know the UK system well enough to know what type of access care biases there may be in that data set. Certainly is not a super representative population if you want to extrapolate to the US, but I'm not sure in terms of socioeconomic status whether there will be a huge bias. Then we looked at all of us, and we calibrate reasonably well in all of us compared to UKBB. But if we were to apply this to, say, specific HMOs and train there, I think we'd be vulnerable to potentially much bigger biases.
also timeliness of, not just, whether you access care ever, but when you.
Sure, absolutely. For cancer, that's really critical. I mean, you know that a lot of the differences in breast cancer outcome, for example, by socioeconomic status of race in the US are driven by access to care, geography. So, you know, yeah, absolutely.
Is there a way?
And I think for the cardiovascular,
Is there a way out of that, or?
is there a way out of that? That's a really interesting ethical question about the extent to which socioeconomic status and race should be part of AI tools like this. Because in a way, if I know that someone lives in rural Alabama, I know that when they're showing up with breast cancer is more likely to be at a later stage. And so should my model know that? Probably in good hands, you know, in the hands of someone who is only intending to make good use of that information, yes, but the potential for abuse is also pretty substantial. So I'm not sure what the answer is. We've been struggling with this, and actually I've seen society struggle with this quite a bit and go back and forth. Like, for example, BRCA1 and for BRCA1 and BRCA2, changing the subject slightly, not too much, is there are founder mutations among the people of Ashkenazi Jewish origin that put them at much, for whom, so if you know that someone is from that origin, you know they have a much higher allele frequency of these mutations. And so the whole prediction paradigm changes in terms of the Bayesian model simply because of a much higher prior probability of being carriers. And over the years, we've always stratified by that. And because we couldn't ignore a variable that gives you a 10-fold increase in probability. At the same time, there have been times where there's been a pushback on using any information about ethnic background for this type of prediction, and for good reasons. So it's really a dilemma that I haven't quite figured out a way of resolving, other than engaging the folks who are delivering the decision supports to, and to the extent feasible, also the people who are receiving the prediction, to involve them in the process, right? I mean, one day we had a laboratory just on this question. Should we not have the Ashkenazi variable in the model? Is that a sort of a way of discriminating? Or should we not have, we have a different baseline for people of Asian origin as well, because the baseline is lower for some of the cancers that we can see. And the consensus of the sort of the young folks who are sort of looking at this with eyes that are not biased by years of convention was that the answer should be in the hands of folks who receive the information, that we should do the model both ways, and that a user based with a patient-based interface could. That's a pretty good answer to me. It requires a level of literacy that is frankly unrealistic. So maybe there was a little bit of idealism in that answer, but at least ethically, I resonate with that way of thinking about it. So going back to your question, circling back to no, it was your question. Can we ever account for that? Accounting for that requires really addressing face-on these dilemmas, and I think. If they have one answer to that, is that that's not just in the hands of the developer. It should be some sort of partnership of the folks who are doing the work that I've presented, the folks who are going to be using the decision support system in their clinical world, and ideally some representation of the patients who are going to be, making decisions based on those. A bit idealistic. It's a real serious ethical dilemma in my mind, and one that a lot of AI faces right I don't know if you follow the controversy about models that use African American status to give different prediction for the probability of renal failure given certain interventions. I mean, I'm not a nephrologist. I don't know anything about that model, but apparently that model was incorporating some pre-substantial biases of calibration, whereby blacks were getting less access to certain important procedures. And maybe 20 years ago, when the model was developed, there was a purely statistical reason why that made sense for that specific data set, but that kind of went away, and the model lost that calibration over the years, and it became a source of bias. Those are things that we need to watch out for. Anyway, long discussion. Great question, though.
And ideally, I guess that improvement in calibration there would provide increased access to care in the context of risk prediction and eligibility.
Yeah, I mean, in general, ideally, anything that you can do, that you can learn about someone to help them understand their risk of being in trouble in the future should help. But it has to be in good hands for that to be the case. Right? Otherwise, it could easily lead to discrimination.
Yep.
I have another question about, you mentioned earlier that genetic testing is costly. Insurance companies don't always cover it unless you demonstrate a family history. So is any of this being used to change sort of formularies of what insurance companies will cover? If you can demonstrate there's an increased probability of being a carrier of something based on family history or other information.
Yeah, so most of the guidelines for genetic testing include use some of these models that I've described. Ours as well as other forms. There are, for each of the various syndromes, there are 2 or 3 models available that work by, been at this long enough that the model is kind of convergent, similar prediction performance, and they're part of the criteria. I'm hoping that by broadening the number of cancers that we include, we'll be able to raise the probability for more people.
Well, right?
Yes, that's right.
I had a question, since a bunch of epidemiologists here, that you showed some plots of probability of disease as a function of patient age that went up, 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 and then down. And so if I was understanding them right, I thought of 2 possible reasons for that. The obvious one, which is competing risks, where you're more likely to die from something else as you get older. And then the other one being selection bias, where if that was important, you would have died from it already. And so I was curious about, I could imagine there are some instances where you could see that apart, but does it matter?
It's a good question. So the model to some extent does that. Because those curves were the importance of each diagnosis in the definition of the signature. The signature is a concoction of those, and so you can think about the whole signature as going up and then down.
Yeah.
But, and so to the extent that all of the signatures at some point go down, then that's a different situation from a situation where a signature goes down before another, when then the risk is a competing risk because the other signature has gone up, and then you see less of the one that is good. If they're all going down, then it is this issue of not living long enough to experience. So to some extent, the model already addressed the question. We never looked at it quite from that perspective.
It was a state model, right? And with, a little bit different time.
Should we call it a? I think you could reframe it as a multi-state model, but the number of states would be astronomical if we want to write it down that way. Our old MS gene is a multi-state model. Yes.
It's a Bayesian network, neural network. It was sort of looking at this what you were describing reminded me of this paper. It was basically saying like, there are these syndromes that tend to co-occur either,
Yeah.
maybe over the course of somebody's life, not necessarily all at once. And I don't think it was necessarily doing the sort of prediction. It was more just like, hey, look, these things
Yeah.
cluster. Like,
Yeah. Yeah, there is work in that space that we have taken as a jumping off point.
So that seems to be more about
Yeah.
like a latent
Right.
variable.
Yes.
Oh, look, there's this,
Yeah.
these things that.
Yes, there are these topic models that David Bly. I'm looking at the wrong cardinal, but
But I do remember conceptually, though, I think in one of our classes we talked about how we name diseases based on the symptoms already after they have that cause that is branching out to multiple diseases later
Yes.
on in all kinds of trajectories. So even though we have all these names for all these diseases, that's just nomenclature based on symptomology and when
Yeah.
you come in and all of that. That was
Yes.
how to think about disease and
but it is a really good point. I mean, this is entirely dependent on the phenotypes that we have, which are going through the ICD codes and they're mapping into the phi codes that we use for training, which also include treatment to some extent. So standard of care is baked into this. When you look at a disease signature, you look at at a history that includes the disease and how it is treated when certain diagnoses come up, particularly cardiovascular diabetes. It's not a natural history model by any stretch, which makes the causal, overlaying the causal part a little bit more complicated. In the Mylynch model, we can say, okay, if you don't do anything about your colorectal cancer prevention, this is what happens. If you do annual colorectal screening, this is what happens. Because we have data on populations where the intervention didn't take place at all. While here, that disentanglement is a lot harder. It's still feasible, though, if there is a new potential intervention and you have a reasonable idea of the causal effect of that intervention in a specific subpopulation, to superimpose that to the risk prediction. Say, okay, this would be a risk based on this model, based on standard of care and everything that is going on right now. If that's comparable enough to the control group in the trial, and then you have a causal effect to put on top of that, you could apply 10% reductions. Yeah.
just wondering, like, with administrative data, it's very difficult. So you would need finer level data to
Yes.
get cancer diagnosis.
That's fair, yeah. That was in part one. I think there was a question about whether the multiple cancer model, I think you're talking about the FAM3 Pro model, the one where I was showing a table of genes by cancer, whether that is trained using healthcare record, and the answer is no. That was still part of what we trained using the genetic epidemiology literature, one cancer by gene association at the time.
Any more questions? I don't know if we've managed to use almost all of the time. So I'd just like to thank everyone. This has been a really wonderful discussion. Thank Giovanni for a really full, accessible talk. Thank you to all of the folks who attended online. There were up to 30 talks, so well-attended talk. Right. If there are no more questions from online, you've had your chance. We're going to sign off now. No, don't sign off. Okay. Thank you.
Thanks, everybody, and thanks for all the questions. Really, really nice discussion.